All right, hello, I'm Amber Rose, and today we're going to discuss energy metabolism in the brain. This is as a precursor for the journal articles we're going to discuss afterward. So, from the very beginning, how do we get energy within the brain? So, production occurs, just as we all remember back from biochemistry. Adenosine triphosphate, or ATP, is synthesized from ADP in both the cytosol, and that's in the process of glycolysis, and then also within the mitochondria via oxidative phosphorylation. ATP is consumed during work that the cell performs. Glucose is the primary energy substrate of the brain and can be metabolized aerobically or anaerobically. Under normal circumstances, about 95% of the energy requirement of the brain comes from the aerobic conversion of glucose to water and carbon dioxide. There's the formula right there where you see that we get a total of 38 ATP. 2 of those ATP molecules come from glycolysis, and then the remaining 36 from oxidative phosphorylation. However, under conditions of hypoxia, there's conversion of glucose that takes place by anaerobic glycolysis, which is in the cytosol, and that's far less efficient. As you see there, you only get a total of 2 ATP as well as 2 lactate. So here's just a little refresher from biochem as well. This is the cytoplasm where glycolysis is taking place, and you get your little 2 total ATP, net ATP that is, and then in the conditions of having oxygen available, it goes into the cell and forms oxidative phosphorylation via the electron transport chain after the citric acid cycle right here. And then overall, you get a net of 36 ATP for that process. So 36 plus 2 is 38. And again, this is aerobic respiration. Oxygen's present. Now, if oxygen is not present, or if there's something else going on, such as in traumatic brain injury, or in the case of certain brain tumors, the brain tissue will instead opt for anaerobic respiration, which is not nearly as efficient. Again, as you see, you come out with 2 net ATP and 2 lactate at the end of that. So when the cell has to decide which way to go, this is the glycolysis, which takes place in the cytoplasm. And then once you get to the step of producing pyruvate, if oxygen is present, then you go on to the Krebs cycle, also known as the citric acid cycle. Or if there's no oxygen, then you either go to anaerobic respiration. In mammals, you have the lactate formed, or in bacteria and yeast, you end up forming ethanol. All right, so why do we care about this? Because, like we were saying, in certain settings, it changes. So in the case of certain cancers, you have what's called the Warburg effect. This is the observation that cancer cells exhibit glucose fermentation even when oxygen is present to properly respire. So that means that even though it could go on to oxidative phosphorylation and go on and produce those extra 36 ATP, it doesn't. It stays in the cytosol and does anaerobic respiration. This mechanism is not understood at this point. However, many studies postulate that the cancer cells shut down the mitochondria due to this particular organelle's function within apoptosis. So basically as a measure to stop the cell from killing the cancer cell. So here's an example of GBM. So you have normal brain cell. It has the cytosol forming the glycolysis process. You get to pyruvate. Now you're moving into the mitochondria, and you're going into oxidative phosphorylation. And you have a little bit going off into anaerobic glycolysis, but very little. The majority is coming this route. Now in the case of GBM, here you are in the cytosol going glycolysis, but even though you have oxygen, the energy production, instead of coming from here, it's going aerobic glycolysis, which means that it's the anaerobic process in the presence of oxygen. It's just not using the oxygen. The oxygen's hanging out but not being used. And here's another image that goes over this Warburg effect. So again, here's regular tissue going to oxidative phosphorylation, and you have a few going under anaerobic glycolysis. But in the case of having this tumor cells, Even if you have plenty of oxygen in the environment, it's only going to do a tiny bit the regular way, and the majority is going to go under what is really the anaerobic cycle, but it's called aerobic glycolysis as opposed to anaerobic respiration in this case, because there's still plenty of oxygen around, even though it goes that way. So you see the same sort of thing happen during traumatic brain injury. Again, the mechanism isn't fully understood, but it's seen. In addition to understanding how energy is produced in the brain, you need to now consider how is energy expended within the brain. So 50% is used for neurotransmitter production, release, and uptake. This is all your synaptic activity. 25% is used for the maintenance and restoration of ion gradients across the cell membrane. And then 25% is used for molecular transport, biosynthesis, and other unidentified processes. So although the glial cells, those are the support cells to all the different astrocytes and so on, although the glial cells account for 50% of the brain volume, they have a much lower metabolic rate and account for less than 10% of the total cerebral energy expenditure. Compared with other organs, the metabolic demand of the brain is high. Brain accounts for only 2 to 3% of the total body weight, and it does not do any mechanical work like the heart muscle, for example. It's constantly beating. Brain doesn't have any actual work, no muscular activity, yet it receives 20% of cardiac output. So glucose and glycogen stores within the astrocytes are extremely limited. So now we're talking about the energy availability to the brain. And there is no significant storage capacity for oxygen within the brain. That's why it needs that 20% cardiac output, because it's not able to store energy. It requires a constant flow of substrate, primarily glucose, to go to the brain. Or there are other substrates, lactate or ketones can also be used, but it's not nearly as efficient to break down. The brain depends on continuous blood flow to meet this glucose and oxygen need. And again, substrate availability is determined by 3 factors. So we just talked about how 20% of the cardiac output gets to the brain. So the concentration of substrate in the blood is one factor of that, the actual flow volume of that 20% cardiac output. And then finally, the rate of substrate passage across the blood-brain barrier. So what happens during traumatic brain injury? So during periods of increased activation, the brain increases production of ATP via glycolysis, increasing lactate production and exhibiting an increase in the cerebral metabolic rates of glucose. Again, increasing lactate production. So remember, even though there's oxygen present, it's going down the pathway of what would typically be anaerobic respiration, but it's called aerobic glycolysis because oxygen is present, but it's still going down that pathway to be less efficient and only produce the 2 net ATP and lactate in addition, or in place of doing the possible 38 total ATP. So non-oxidized glucose may also be consumed in the pentose phosphate pathway, which is another pathway for generation of NADPH needed to replenish glutathione and dampen the oxidative stress. And this is used for synthesis of neurotransmitters and structural components of all cells, or it could be otherwise converted into lactate, and that's the predominant form that comes about. So there's a lot of lactate production in traumatic brain injury is what your take-home point is from that, and that's what the brain is using a lot of as its substrate, as opposed to primarily glucose, like in the normal state. So does that mean that lactate is a better substrate in traumatic brain injury? So studies have shown that glucose sustained mitochondrial respiration at a low level, fixed rate, but increasing its concentration did not result in upregulation of oxygen consumption. So what does that mean? It's saying that when normal states are in place, that the mitochondrial respiration rate of just going through the normal glycolysis in the cytosol, followed by the citric acid cycle and the electron transport chain within the mitochondria, is just done at a fixed rate, just normal, just like an assembly line. However, lactate administration resulted in a dose-dependent increase in mitochondrial oxygen consumption that may well have enabled the mitochondria to meet the increased energy needs of the injured brain. So it's saying that with that fixed rate, that the mitochondria, being that assembly line, just uses just as much as of the oxygen as it normally does, just around the clock, no more. But it's saying that by administering lactate as a substrate, resulted in the mitochondria consumption of oxygen that is actually less. And so it's saying that even though it's a less efficient way to produce energy for the cell, that the mitochondria are requiring less oxygen to produce that energy. So again, this is not well understood, but this is what they're noticing within these studies. That might be a reason why all this is happening. So going back to cerebral blood flow, because this is something that we can manipulate in our patients. So as a refresher, cerebral blood flow is regulated to provide an adequate supply of substrates to the brain via changes in the arterioles. Normally the diameter ranges from 30 to 300 micrometers. Autoregulation occurs in response to metabolic signals, blood pressure, viscosity, and changes in the arterial partial pressure of carbon dioxide. So going to that first bullet, metabolic autoregulation. So in general, the brain responds to alterations in metabolism by any changes in flow. So increased metabolic demand is matched by a proportional increase in cerebral blood flow. For example, fever, traumatic brain injury, it's going to increase your metabolic demand. So ideally, you should have a proportional increase in cerebral blood flow to meet that need. Decreased metabolic demand results in, similarly, a diminished cerebral blood flow. So for example, if somebody's under deep anesthesia, their metabolic demand in the brain goes much lower. So their cerebral blood flow is then decreased by changing the alterations in flow. Also deep coma, same thing happens. Now the second bullet that we had mentioned earlier, pressure autoregulation. So the blood viscosity and rheology, which is the actual flow of the blood, the flow of the fluid, is affected here. So hemoglobin goals are not clearly defined in the literature for traumatic brain injury. However, we all know that hemoglobin goal of greater than 10 is accepted by many institutions. However, it is not 100% accepted by all institutions. And that's controversial, it's another issue. But for our purposes, in our program, when we have a patient with traumatic brain injury, our goal is greater than 10 for hemoglobin. Cerebral perfusion pressure is the mean arterial pressure minus the intracranial pressure. And for example, an average person with a MAP of 65 to 90 and an ICP of 5 to 10 millimeters of mercury has a CPP, that's right there, range of about 55 to 85 millimeters of mercury. For our purposes for trauma, we ideally want to have somebody around the range of 60 to 80 millimeters of mercury to ensure that they have adequate cerebral perfusion pressure. Those are the references. Any questions? That's pretty dense, I know.